So I think that we can start. Thank you very much to the speakers that will be in this panel. First of all, Lidia Strigari from the University Hospital of Sant'Orsola in Bologna, Italy, and Professor Christopher Porada from the Wake Forest Institute for Regenerative Medicine. Let me thank also the Policy Studies Organization to give us the opportunity to present this panel about advancing in space radiobiology through interdisciplinary research. Insight from the MS Roma Sapienza group, because since 5 years, this is the kind of research that we are doing in collaboration with the Lydia research group. So let's start with the panel. This is the agenda. I will introduce the alpha magnetic spectrometer experiment on the ISS, taking cosmic ray data since 2011. And AMS is the acronym. And then the group, that is one of the group in the collaboration, the Roma Sapienza group. And something about the ERCCS, Azienda Ospedaliera Universitaria Policlinica di Sant'Orsola Bologna, that is in this kind of research since 2017 with us. And as an example of interdisciplinarity research, I will talk about a topic collection that we launched in 2022 to try to create a networking on space radiobiology and in general with other people working on the same topics. And this is will be the turn of Christopher Porada that take part to this topic collection, but this is the occasion to share with us the research that is doing at the Wake Forest Institute for Regenerative Medicine. And if there will be time, we will have a short roundtable about this kind of topics. So alpha magnetic spectrometer, also MS02, is a particle detector measuring galactic cosmic ray fluxes. It was a very, very complex experiment. It was installed on the International Space Station on May 19, 2011, but to build it, it required more than 15 years with a big collaboration, international collaboration, in which we are a small group inside of many others. The lead is done by the MIT in USA, and especially the spokesperson is a Nobel laureate in physics, Sam Ting. And of course, for all this kind of activity, there is the needs of the strong support from the NASA for the operation. I mean, so what you see on the right is the control room of the experiment, in which there is the control of the experiment and the monitoring of the data and of the telemetry, and is operative every day of the year, 24 hours per day. It's located geographically at CERN in Switzerland, but is in contact with all the main control room of NASA in the US. To now, the detector is it's taken more than 205 billion cosmic ray events. More information, you can have it on this website here. And the real point is that this kind of science data taking is done to investigate fundamental physics open problem. From a point of view of, let me say, why MS is suspicious is the fact that it was able to measure all the three components that compose the space radiation that are present in the heliosphere, the so-called galactic cosmic rays, the part that comes from outside the heliosphere and are generated by the astrophysical sources. The cosmic rays, instead, they are generated inside the heliosphere by the sun. And the last component, the third one, that is very important for space radiation, for space exploration especially, are the one that is trapped inside the magnetosphere, the so-called Van Allen belts. And the typical measurement of the MS detector is essentially done by the fact that it is composed of many different instruments. It's not just a single instrument. And as you see from this table, on the road, you have the instruments with the acronym that characterize the instrument itself. But the real point, the main point, is the fact that each single particle that constitutes the cosmic ray, electrons, protons, nuclei, will leave different signatures inside this instrument. And by combining all the information collected by the different instruments that compose the MS, it is possible to reconstruct not only the type of particle that go through and so constitutes the cosmic rays, but also some important characteristics, like, for example, the momentum, the energy, the charge, and so on. For example, this is the typical reconstruction, the periodic table that constitutes the cosmic rays, starting from helium up to very heavy nuclei, as reconstructed by the MS. The typical results, in that sense, are essentially of two types, to be very, let me say, in easy description. Absolute fluxes, that means that we are able to measure the fluxes. That means for each isotope of particle, the number of particles that per second arrives in the MS, so in the near Earth field. And since the data taking starting, that was in 2011, we did a lot of publication on the physical review letters of electrons, protons, many other type of particles. This is an example of the last one. And you see, for example, in this publication, there is the flux from the very low energy to higher energy of the sulfur, that is one of the components of the cosmic rays, in comparison with other nuclei, like, for example, neon, carbon, magnesium, and so on. Another very important for space exploration and space radiation characterization is the fact that the MS is taking data since 2011. So at the moment, it was able to not only give us information about the absolute fluxes, but also how they behave in time. Because of course, not of course, but there is the galactic cosmic rays are not constant, in the sense they are modulated by the activity, magnetic activity of the sun, and so they appear in intensity differently with respect to the solar cycle, so with respect to how the sun behaves. And this is important because, of course, the energy is correlated to the possible damage that this cosmic discharge particle have on the biological humans. And so this could be, of course, important to calculate this kind of things. For what regard the activity that we are doing in Roma since 2017 is an interdisciplinary one, because we put together, we mesh up these three components. The fact that we are since many years in the SMS2 collaboration, so we have really expertise in the cosmic and space radiation. The fact that this radiation, of course, is not only important for fundamental physics, open question like dark matter or things like that, but also is important because it's exactly the same radiation that will be at which will be exposed the astronauts, so in the future, the space workers, if they go into the space. And this is combined also with the fact that in the last 5-10 years, there will be a restart of the human space exploration, that after the Apollo mission was confined in the LEOs, where the radiation is not so concerned, but now there is a lot of activity to have again the humans go outside the low Earth orbit and go to the moon and then to the Mars. For example, last year there was the first Artemis mission that for the first time have give the possibility to a starship to fly in the nearby of the moon. So the activity was done in collaboration, as I said, with the Policlinico Sant'Orsola, that is the medical division that is now here represented by Lydia. While on the left, you see where we are located. Our group, the MS group, Roma Sapienza, is of course in Roma, in one of the Department of Physics building. And while Bologna, that is another Italian city, not far from Roma, 400 kilometers to the north, where Lydia is instead leading the medical physics department. So this is the group, because we would like to present the person. We are, of course, person in Roma, person in Bologna, but we also have open since many years the possibility to collaborate with many other people. For example, last summer, we have a summer student for the University of New Mexico Albuquerque that stands for us, stay with us for 2 months, thanks to an agreement between the INFN and the DOE, the American DOE. But also we have collaboration with many other people and researchers. Let me say, we try also to have international collaboration, for example, with neuroscientist Feng Tang in National University of Singapore. We are now another collaboration to just make something together in this field with the group that in the National Institute for Laser and Plasma Radiation Physics in Romania, and so on with other institutes in Italy. Especially we are with this last institute that I mentioned, for example, evaluating the possibility to use the laser plasma accelerator as a novel versatile tool for space radiation studies. Because in the future, there will be the need to test also in the radiobiology samples more than in the past. And so maybe the standard linear accelerator technique could be complemented by using these accelerator techniques that starting from plasma focused laser beam that its specific target create different type of particle then can be accelerated with the standard techniques. The important thing is that, for example, from a single target, it's possible to generate at the same time different kind of particle, like for example in this figure, protons and electrons at the same times. And this is important because one of the point of the space radiation, with the example with respect to the clinical field, there is no single isotope or mono-energetic isotope that is, or particle that is used, but instead the particle altogether and with different energies. This is one of the characteristics that make the space radiation problem so complex. So other collaboration with other university in Italy, always in this kind of space radiation field. The start of all was, of course, done in 2019, but we started to work in 2017 when we published the first work in which we show in collaboration with Lydia that it was possible to use the data taken from the astroparticle experiment to improve in the radiobiology because they contain all the information that they need. For example, we published this galactic cosmic ray sensitivity analysis in which we show that most of the particles that MS collect are essential, useful for this kind of study. And also, we make a publication in 2021 in which we show how this kind of data could be used in different parts of the space emission risk assessment process that is very complex one. Also, we started the collaboration with the scientific community of different type, radiobiology, medical physics, radiotherapy, nuclear medicine. We try to start, of course, to talk with people that works in what is relevant for the space exploration, especially the space agencies. And we continue, of course, to do this kind of study. One also, let me say, what's initiative was to launch in 2022 a research topic collection that, in a sense, is just an editorial initiative. But before this, I would like that Lydia will describe the activity that she did with this group in Bologna, and also the activity we did together the last year. I don't know, Lydia, if you want I go on with the slide, or you want to?
No, you can go on with the slides. It's fine for me. Okay. So thank you, Alessandro, for the introduction. I just present my institute. Is one of the largest public institute in Italy. That is a research institute dedicated to do translational research, also based on several diagnostic and therapeutic applications that we perform day by day. And the idea is that when we try to understand the dose effect relationship based on our clinical experience, we take into consideration the citizen or patient presentation, the type of irradiation that is mainly rely on diagnostic use when we have some screening or investigation with imaging and so on, or patient undergoing, for example, treatment. And this is the type of exposure that we are looking for. And then the last aspect is the type of endpoint that this machine and treatment and irradiation may produce in our patient. So the idea is just to take care of this information, because we have a lot of patients, more than 1,000 million of information collected day by day. And so the idea is just to try to have this information as a strategy to take into consideration the citizen sensitivity, the gender effect, the body mass index effect, and so on, and have to this information just to characterize the citizen and the patient that is just a large population compared to astronauts that are healthy citizens, obviously. Alessandro, if you can go on, we'll just have a look to the fact that we can have not only therapeutic application as reported in this slide, but we have also several combined treatment that we several imaging modalities available, and also treatment with nuclear medicine therapy, in which we use several types of particles. So we have electron, photon, but in general also emission of fluoride-19, generate antimatter, and so on. So we have several types of way to produce this irradiation in the patient, not only in specific areas, but also in the whole patient during this type of examination. If you can go on. So the idea is just to take into consideration that there are several dose effect models that have been already established based on Earth experiment to mimic space exposure. And we have also dose effect models that are obtained based on an accurate precise absorbed dose estimation for several types of endpoints. So the idea is just to identify in this paper the type of irradiation that are usable for understanding better the dose effect models, the type of data that are available, and the actual mechanistic or phenomenological model that are available. They try to have a benefit integrating multiple types of experiments based on in vitro study, preclinical study, clinical studies. And in this way, we can try to combine data and model and apply this information to predict and produce a more robust dose effect relationship. If you can go on. So just as example, we have a look to several types of exposure and effect, for which we have a dose effect model for eye flashes, cataracts induction, cerebral and cardiovascular disease induction, obviously cancer, and several modulation of biomarker, protein, or measurements of chromosomical aberration. This has been already investigated to those ranges that are typical of diagnostic and irradiation with therapy at the first fraction, for example. In this paper, we review the number of models that are available, and we try to verify if these models are in agreement with each other, or if we can have a benefit when we combine the dose effect model derived from a very high number of citizens and patients with a model derived on few astronauts based on the limited time exposure that we have in the past. Again, if you can go on, we try also to investigate a very nice topic in dose effect model in clinical preparation that are the non-target effects. So we know that the direct effect on DNA is just one of the possible way to use this apoptosis and death of the cells, but there are also several possible indirect effects that should be considered. There is also an interaction with the immune system of the citizen, of the patient, that will be taken into consideration when we try to reduce the possible effect of radiation, or when we try to have a benefit for the patient treated for a given disease. In this case, we have a look to the possibility to improve the model of non-target effect based on experimental data and try to refine the models to describe this effect. Again, these non-target effects, so at the moment are based on experimental data that are limited to in vitro models and in vivo model. But we have also the opportunity to try to enlarge the knowledge from these and improve the knowledge from this effect, taking into consideration the accurate and precise characterization of the space fluxes based on IMS data. And this means that we can just try to predict the tumor prevalence, and we can try to distinguish between the type of direct and indirect effects that are generated to produce these secondary exposure, this secondary effect after an irradiation with ionizing radiation. So in our vision, we could expect it to have an evolution because we have obviously a lot of information to be combined in some way. And the idea is to just try to use AI-based models to collect, to combine clinical data and try to produce phenomenological or mechanistic models that could be useful to explain the experimental data that are collected day by day. So this could be one of the possible strategies to improve this presentation. Alessandro, you can go.
Yeah. Okay. So thank you very much, Lydia, for taking the duty to talk about the research that we did in the last 5 years, or part of the research. So now is instead about the interdisciplinarity. I would like to talk about this research topic initiative that was done on the Frontiers publisher, and it was launched at the end of 2021. The idea was to try to involve, create collaboration in this kind of field. I have to say that Frontier were really enthusiastic to support this kind of research topics. And as you say, that it was possible to publish on 3 different type of journal: astronomy and space sciences, physics, and public health. This is let me say, the witness of the fact that there is this kind of research is really interdisciplinary. Another things that is, of course, we try to do is to, let me say, try to involve in this kind of research as much as possible different, also starting from the editorial board, the kind of people. So people that works in the high energy physics, particle physics, standard 1, people from the medical clinical fields, and also, of course, from the biologists. And for example, one of the person that was in the board was Nan Ding from the Chinese Institute, the Institute of Modern Physics of the Chinese Academy of Science. It was a biologist. So there was Lydia, and there's Gianluca Cavoto that was instead involved for Sapienza University in experiment essential on, let me say, fundamental physics, and Cristina Consolandi from the University of Hawaii. Also, she's a member of the MS collaboration, but his work is focused on the low energy part of the space radiation, the one that is most important from the, let me say, space radiobiology works. So the research topic, as this was really interdisciplinary. This, let me say, it's the result of the fact that it was accepted by 3 different Frontiers journal. And also we received some interest, at least from very different scientists. You see, this, I try to do this kind of, let me say, taxonomy or classification in radiation detection, so physics, nanobiology, medical physics, and radiotherapy. Scientists like space science, radiobiology, particle physics. That was equally distributed in the, at least in try to collaborate or try to send have interest on that. At the moment, after 2 years, we have more than 7,000 views for the topics and for the article that was published on the research topics, more than 1,000 downloads. We received more than 5 abstracts. At the moment, the 3 was published with more than 30 authors. Of course, if you want, you can still apply. This is the QR code. But let me say that it's now time, since he was so kind to participate to this panel, to give the word to one of the scientists that take part of the research topic, Professor Christopher Corrada. You can share your screen, Christopher.
Thank you. Yeah. Okay. Is that projecting correctly?
Yeah.
Okay, thank you. So thank you very much for the opportunity to be a part of this panel and to talk a little bit about the research that we've been doing for about the last 8 years, I guess, at the Wake Forest Institute for Regenerative Medicine, focusing on trying to define the biological effects of solar energetic particle and galactic cosmic ray radiation on the hematopoietic and the gastrointestinal systems. So as probably everyone certainly on this panel and perhaps viewing the panel know, scientists and regulators have identified that space radiation is probably going to be one of the largest factors limiting prolonged missions in deep space, such as the ones planned to the moon and to Mars. And as we heard briefly, space radiation is quite a bit different than the types of radiation that we're used to on Earth. We have 2 different things to worry about. So we have solar energetic particles, or SEP, which are primarily protons released from the sun. And the important thing is that these are released intermittently, and they're unidirectional since they come from the sun. The more troubling type of radiation would be the galactic cosmic rays, or GCR, which are also mainly high energy protons, but they also contain high atomic number and energy, or HCE particles, which are atomic nuclei that are atoms stripped of their electron shell. And the problem with GCR is that it originates outside the solar system. It's continuous and it's omnidirectional, so it poses a much greater problem from the standpoint of shielding and trying to protect the astronauts. Unfortunately, we don't really know a lot about the biological effects of SEP and GCR radiation, and one of the main reasons for this is that when we're on the Earth, quite happily, almost all of the SEP and GCR is deflected as a result of the magnetosphere around the Earth that acts as a shield. And happily for the astronauts on the ISS, you may think, well, they're not on Earth, so perhaps they're exposed to a lot of these types of radiation. The ISS actually orbits at an altitude of only about 250 miles. The magnetosphere extends out to about 40,000 miles above the surface of the planet. So even the astronauts on the ISS are not exposed to very high doses of GCR radiation. And one thing I thought was, well, at least the astronauts that were in the Apollo missions that went to the moon, they were certainly well outside the magnetosphere. So they must have been exposed to some GCR, and maybe we could get some data on what happened to them as a result of that exposure. But it turns out the moon spends about 6 days every month inside the Earth's magnetotail, which you can see here in this yellowish tail, basically projecting to the right of the Earth. And it turned out that NASA engineers were brilliant or insightful enough to realize this, and they planned the Apollo missions to coincide with the time of the month when the moon fell within the Earth's magnetotail. So even the astronauts that were in the Apollo missions and were on the surface of the moon were still largely shielded. We're not exposed to particularly high doses of GCR radiation. So we really don't have any data on humans that have been exposed to this type of radiation, which makes it very difficult to assess what the risk may be to astronauts spending a long time on the moon or going to Mars. So we initially decided, well, since this is one of the focus areas of our lab is the hematopoietic system, but to focus on the effects of SEP and GCR radiation on the human hematopoietic system. And we thought this was a particularly important system to look at because the hematopoietic system is extremely radiosensitive. Damage to this system could be life-threatening in a very short time due to the need to replace roughly 5 to 6 billion cells every day. Also, leukemias are one of the most frequent radiogenic cancers. And importantly, from the standpoint of looking at mission success, leukemias have a short enough latency that if we're thinking about a planned mission to Mars that would take 3 years or more, their latency is actually short enough that they could arise and progress sufficiently to compromise a mission. So just a little bit of a refresher. Hematopoiesis, basically, if we're looking at all of the different mature blood and immune cells that are circulating within the body at any given time, all of those cells are ultimately derived from a very small, rare population of cells that are within the bone marrow of an adult known as the hematopoietic stem cell, or HSC. So we initially focused our studies thinking that any impact that SEP or GCR radiation had at the level of the HSC would probably be the most profound, as it would impact all of the lineages that are derived from that stem cell. So we performed studies at NASA Space Radiation Lab, or NSRL, where we took bone marrow from healthy adults who were of typical astronaut age, isolated the hematopoietic stem cells by sorting for CD34 expression, brought these cells to NSRL and exposed them to a variety of different radiations. So in this initial pilot study, we used protons to model the primary solar energetic particle or solar particle event. We used gamma radiation at 1 gray as a reference. We brought the cells, put them into the room, and took them back out without exposing them to anything as a sham irradiation. As a model of GCR, we used iron ions. And then based on studies several years ago now by Betsy Sutherland at NSRL, we also exposed the cells sequentially to protons and then iron, as she had shown, at least in human fibroblasts. If you expose cells sequentially to protons and then to GCR ions, you actually have a much more pronounced effect than if you expose them to either one individually, or interestingly, if you expose them to those same 2 ions, but in the reverse order, so hitting them with iron first and then protons, you don't see any synergistic effect. Once we had done that, we then brought the cells back to Debuform. We did studies looking at chromosomal aberrations. We studied colony-forming potential of the hematopoietic cells. We did DNA and RNA analysis. And then we transplanted these cells into conditioned immunodeficient mice so we could study what happened to hematopoiesis in vivo as a result of the cells having been exposed to modeled space radiation. And this is just kind of a schematic of how we use this in vivo xenograft model to study human hematopoiesis in vivo. So basically, we irradiated the cells, as I said, brought them back, conditioned the mice, and transplanted them with these cells, and then waited for about 5 to 6 months after the human hematopoietic stem cells had repopulated the host, and then looked at the presence and levels of human cell engraftment, differentiation, and lineage commitment, and looked to see whether any pathology is developed. The first thing we noted is that as a result of exposure to different types of radiation, so gamma, proton, iron, and then mix here is the proton followed by iron, we saw that there was a marked skewing of lineage differentiation of the human HSC in vivo as a result of these exposures. So we had an expansion of the T cell population at the expense of the myeloid and the red blood cell populations within these mice. The most profound result we saw was that in mice that were repopulated with HSC that had been exposed to a Mars mission relevant dose of iron ions, many of these mice had tremendous splenomegaly. So at the top, we can see the spleen from one of the sham irradiated mice. And on the bottom, and note that we have the ruler at exactly the same scale. This is the spleen from one of the representative mice that was repopulated with HSC exposed to iron ions. So the spleen was about 30 times the normal volume. Importantly, when we went and we stained with human-specific antibody to CD59, we showed that the entire spleen was basically packed with human cells. So the splenomegaly was caused by some kind of abnormal proliferation and expansion of the human cells within the spleen of the mice. We then worked with a team of clinical hematopathologists at the Wake Baptist Medical Center who went through and did basically exactly the same clinical workup they would do for a human patient that came in presenting with some type of hematologic malignancy, and diagnosed that these mice had actually developed human T-cell acute lymphoblastic leukemia as a result of the human HSC having been exposed to iron. So to our knowledge, this is the first and only human cancer that's ever been reported as a result of exposure to GCR radiation. We also did whole exome sequencing on the HSC that had been exposed to the different radiation types. And you can see in the Venn diagrams on the left here from the 2 different donors, we had very similar patterns as far as how many different mutations were being induced and what types of mutations. as a result of exposure to each of the different ions. The interesting thing we can note is that there are many mutations that are common to all of the radiation types, but then each radiation type also has its own unique population of mutations that were induced. One interesting finding, which we still don't have an explanation for this, and we'd be welcome to any suggestions as to how we can figure out why this may be occurring, but we found SEP and GCR radiation seem to frequently produce mutations in genes that had been implicated in leukemia or involved in the process of hematopoiesis when compared to gamma radiation. And we think this may be why we see leukemia when the cells were exposed to iron ions, but we're not really sure why space radiation would have some proclivity for mutating genes that are involved in leukemia or in hematopoiesis. And we reported these findings in leukemia. And we also had another paper that came out looking at biological bystander effects, where we showed that if we exposed cells of the bone marrow microenvironment to SEP or GCR radiation, and then placed normal unirradiated human hematopoietic stem cells in close proximity to the microenvironmental cells, we actually saw alterations in hematopoiesis as a result of these biological bystander effects as well. So one question that arose when we've presented and tried to publish those findings was, well, you took the hematopoietic stem cells, you flew them to Long Island, you exposed them to radiation, you flew them back to North Carolina, and then repopulated mice with those cells. What if the reason you're seeing some of these leukemias is because of all the manipulation of the cells outside the body? It would be much better to study what happens if you were able to expose human hematopoietic stem cells inside the body to these same radiations. So this was exactly what we did. We decided to create what we called humanized mouse avatars. So basically we took our immunodeficient mice, we repopulated their hematopoietic system with human cells, again taken from the bone marrow of healthy donors of typical astronaut age. We waited until their hematopoietic system had been humanized, and then took those humanized mice to NSRL and irradiated with whole body irradiation to a variety of different radiation types. So first, before we brought them there, we looked by flow cytometry just to make sure that we had multi-lineage human engraftment in all of these mice. And so over the course of about 7 years, we went back and forth to NSRL, exposing over 500 humanized mice. And these mice had been humanized with cells from 9 different human donors and with donors from both genders. And we exposed them to a variety of different ions designed to model SEP or GCR. So we used protons, oxygen ions, silicon ions, iron ions, and more recently we were able to take advantage of NSRL's new 5-ion simplified GCR simulator. And once again, like we saw when we had irradiated cells in vitro and then repopulated mice, we saw marked skewing again of the lineage representation in vivo of the human hematopoietic cells, with a marked expansion again with the heavier ions within the lymphoid lineage. We also saw this in the peripheral blood and in the spleen. And interestingly, within the spleen, when we did flow cytometry, we saw that the lymphoid expansion seems to be an expansion of CD8 positive cytotoxic T cells. Importantly, once again, when the mice were exposed to whole body irradiation, once again at Mars mission equivalent doses, we saw a tremendous splenomegaly. So in this image in the upper left here, for those of you familiar with mouse anatomy, the spleen usually is pretty hard to find. You have to kind of dig the other viscera out of the way in order to find it. But here you can see the spleen filling almost the entire abdominal cavity in one of these mice. And this was one that was exposed to oxygen ions. When we did histopathology, you can see we detected these abnormal nodules of cells circled in green here. And importantly, when we went back in, we did immunofluorescent staining with an antibody to Ku80, which is a human-specific nuclear marker. You can see these abnormal foci or nodules are actually comprised entirely of human cells. And once again, down here, we can see the spleen from one of these mice that was exposed to oxygen ions compared to a spleen from a mouse that was exposed to gamma rays. So once again, showing tremendous splenomegaly like we saw when the cells were treated in vitro first and then used to repopulate the mice. Here we went through and actually quantified both the incidence and the severity of splenomegaly in all of these mice as a result of exposure to different ions. And you can see that as we go up in LET, basically we have an increase in both the incidence and the severity of splenomegaly. And importantly, none of the sham irradiated mice with cells from any of these donors ever developed splenomegaly. And only 3 out of, actually I can't even tell how many dots that is, but I think it's probably about 24 or so mice that were exposed to gamma radiation. Only 3 of them developed splenomegaly, and it was very mild splenomegaly. So to kind of summarize what we found with the hematopoietic system, our results to date indicate human hematopoiesis is markedly altered following exposure to Mars mission equivalent doses of protons and iron ions. We've now moved along to look at in vivo exposure, and it appears that this also induces hematologic malignancy like we saw when the HSC were exposed in vitro and then transplanted. The humanized mice have been exposed to oxygen, silicon, and the 5-ion simplified GCR simulator. They have also exhibit marked splenomegaly and pathology that seems to be consistent with hematologic malignancy. And this has now been seen in mice that were repopulated with cells from 5 different human donors. So the next steps, we have a licensed clinical hem-onc pathologist at the hospital who is reading all of the spleen sides and staining with various markers to try to diagnose the nature of the malignancies that are present in the humanized mice exposed to each of the different radiation types and doses. And hopefully within the next couple months, we should have all of those diagnoses and be able to write a paper summarizing all of these findings. One thing that we did do during this process also was develop a method that enables us to isolate the human cells from the humanized mice and obtain a purity of about 99% human cells afterwards. And we're quite excited about this because the ability to pull these human cells back out of the mice after they've developed some type of malignancy as a result of exposure to space radiation will enable us to look at the genetic basis of the GCR-induced human cancer, look at possible gender effects, since we had, as I mentioned, mice that were repopulated with HSC from both male and from female donors. We can potentially look at genetic factors for individual variation in susceptibility to radiation-induced cancer. As from what we've seen so far, not all of the HSC donors appear to get malignancies following exposure. And we're hoping we can also look at the possible LET effect, since we have ions with a variety of different energies that were used for the exposures. And the second set of studies that I wanted to go through quickly, which were actually featured in the special issue of Frontiers, we wanted to look at the effect of space radiation on the GI system, because the GI tract is also extremely sensitive to a variety of types of radiation. Unfortunately, there isn't really any way to humanize the GI system of a mouse, so we were forced to do these studies in wild-type mice. So basically, we took wild-type mice to NSRL and exposed them to a variety of different radiation types and doses, and then euthanized the mice at multiple different time points and performed a variety of analyses to look to see what happened to the GI tract as a result of exposure to these different modeled space radiation. The first thing that we noted was both SEP and GCR radiation induced marked leukocyte infiltration and lymph node swelling or enlargement of the lymph nodes. So you can see here the sham irradiated all the way on the left. The lymph node is almost undetectable. With gamma, it's really not much bigger, but with proton, iron, and with oxygen, the lymph nodes are absolutely enormous and completely filled with lymphocytes. Both protons and iron ions to model GCR also appeared to induce vascular damage and vascular abnormalities within the small intestine. And it was noted that a lot of these abnormalities were seen within the lacteals, which are lymphatic capillaries that absorb dietary fats in the villi of the small intestine. We did staining with Claudin-3, which is labeled in red here, and you can see that exposure to either SEP or GCR radiation led to a marked reduction in staining for Claudin-3, suggesting a disruption of epithelial barrier function. We then wrote a script so we could go through and actually precisely measure the length of these epithelial tight junctions. And when we did that, we saw that with iron, at least, we had a significant reduction in the length of these tight junctions within the small intestine. We also did staining with Musashi and with Chi-67 to look at both the levels of intestinal stem cells with Musashi 1 and then proliferation of both all of the cells and then co-labeling with Musashi to look at proliferation of intestinal stem cells. And it appears, at least, and these are studies that are still ongoing to try to quantitate, but all of the radiation types seem to have impacted proliferation within the intestine. And it seems as though the heavy ions, at least, may have done something directly to the intestinal stem cells, as we see a change in the intensity and the amounts of staining with Musashi within the crypts of the intestine. So while we were doing these studies, our collaborators at UC Davis and Lawrence Livermore Lab were doing microarray and analyses looking at cells that had been exposed to each of the different radiation types and looking for differential gene expression. And when we did this, we found that many different pathways that were being altered by exposure to radiation can be impacted by the dietary supplement curcumin. So we set out to see whether curcumin might be something we could use as a very simple countermeasure to try to protect from these types of radiation. So initially what we did was take hematopoietic stem cells, expose them in vitro to different radiation types in the absence or in the presence of curcumin, treating either before radiation or after radiation. And what we saw was that at least treating before irradiation, looking at colony forming output, giving curcumin prior to exposure to radiation seems to largely restore some of the colony formation loss that you see with each of these different radiation types. So we thought these were promising preliminary data. Unfortunately, curcumin has terrible water solubility, and as a result, it doesn't have very good bioavailability in vivo. So our collaborators at UC Davis and Lawrence Livermore developed lipid nano protein particles that enable us basically to package the curcumin and enable it to become soluble so that we can administer it in vivo and give it to the mice to see whether it provides radioprotection. And what we found, at least looking at the small intestine, and this is tunnel staining, so looking at apoptosis within the small intestine, on the left, we can see 24 hours post-iron irradiation, all of the various cells undergoing apoptosis. And on the right, we can see a representative section from one of the mice that was pre-treated with these curcumin-loaded NLPs 24 hours prior to irradiation, and we see a marked reduction in apoptosis. When we stained for gamma H2AX, so looking at double-strand breaks, we can see on the left that there was a tremendous amount of DNA damage at 4 hours after exposure to iron. And in the mice that were treated with CNLPs, we have almost a complete absence of gamma H2AX foci, suggesting that we may have provided protection against this DNA damage. And the results of this are quantified and shown in graphical form on the right. And all of these studies were published, as I mentioned, in this the special issue that both of you edited on astroparticle biological effects. So one thing I mentioned, it's not possible to humanize the GI system in mice. So one thing that we're working at trying to accomplish at WFIRM are designing OTEs, or organ tissue equivalents, or basically 3D organoids of a variety of different tissues. And we currently have funding from NASA to start using some of these 3D organoids as a way of studying the effects of space radiation and other stressors that are present during spaceflight on actual human organoids, instead of having to use mice as a model system. And here you can see some of the 3D gut or colon organoids that we've developed, staining for a variety of different cell types and looking for function based on tight junction formation. And I'd like to finish just by acknowledging all of the people that were part of the team that enabled the generation of all this data and enabled us to do all of these studies. And finish by acknowledging, expressing gratitude to the various agencies that have funded the work that we've done. So, thank you.
Thank you very much. Thank you very much, Christopher, for this presentation. I try now to share again the screen to let's start the, I don't know, Lydia, if you want to say something on the Christopher presentation.
No, I really enjoyed the presentation. Thank you so much for this very nice overview on your research on the possible investigation on radiation protectors that could be very useful also in practice. I have just a question, if I can.
Yes.
Just to start, what type of exposure you said that you used acute exposure and doses that are similar to diagnostic use or more similar to, I don't know, high dosage just to reveal this exposure, because I know that, for example, for M2, I can say you should use a little bit higher doses to reveal this modulation. So I'm very interested to know about those range that you investigated.
So the doses that we picked were actually one of the people that I acknowledged, Paul Wilson, who's at the Pacific Northwest National Lab, is a radiobiologist who was actually at NSRL and ran the biology program for several years. So he calculated all of the doses of each of the different ions to model exactly what astronauts would see basically during a 3-year Mars mission. The unfortunate part of working with NSRL, and nobody seems to have figured out a way around this so far, is the dose rate. So the issue we have is that the astronauts would basically be exposed to that dose over the course of 3 years at very, very low dose rate. And at NSRL, basically the only way to do the exposures is an acute exposure. So the mice go in and they're exposed to that whole Mars mission relevant dose probably in about 10 or 15 minutes, depending how many different ions you're using. So there's a lot of debate and discussion in the field what that does to the data. So the initial thought was that by giving them the entire dose acutely like that, we may actually be overestimating what the damage is that's going to occur as a result of a Mars mission. But kind of counterintuitively, what Paul Wilson has shown, at least in human fibroblasts, is if he gives exactly that same dose, but splits it up over the course of 2 weeks, for instance, going in every day and giving them a small part of that dose to get the same total dose, he actually sees a much higher rate of transformation in the ones that receive the protracted lower dose rate, getting the same total dose, than in the ones that get the acute dose. And what he likes to say about that is that basically dead cells can't cause cancer. So his thought is if you give the entire dose all at once, a lot of the cells have so much damage, basically they can't survive or find any way to repair the DNA sufficiently to continue proliferating, and they basically undergo apoptosis and die. But by protracting that dose out over a longer period of time, the cells are given time to recover and repair in between exposures. As a result of that, you may have propagation of mutations that can ultimately lead to cancer. So our thinking at this point is that we may actually be underestimating what the true risk of carcinogenesis is by giving the entire dose as an acute dose rate. So one thing we've tried to do, and unfortunately we weren't funded to study this, but Mike Wild in Colorado actually has a low dose rate neutron facility that he's been using to try to answer questions about what would happen if you exposed, in his case, whole animals. He's using mice to basically a Mars equivalent dose of neutrons, but over the course of a year or 2 years. So he has basically a housing facility where the mice are just sitting there with the neutron source, and the neutrons are slowly coming out at a dose rate that would mimic kind of what would happen in deep space. So that, to my knowledge, I think is the only way that that kind of question could be answered with the logistics of NSRL. At this point, there isn't any way to provide a really protracted dose. I know there are some investigators that are going and doing studies on mice with the GCR simulator, that they're going in over the course of like an entire month, exposing the mice for a couple minutes each day over the course of 2 months, but it's still, I mean, it's not 3 years, it's only 2 months. So you could still argue that it's not an exact model to look at that. Yeah.
My question is, obviously, thanks for your answer. My question was about on the fact that we have sometimes some models that should be based for ethic concern and practical concern that are a little bit different from the chronic exposure that is expected to have on the moon, obviously. But I think that your results are very interesting and could be also have a very large impact on clinical practice, also for human exposure and during medical application and so on. So my question was about also on the possibility to translate your knowledge and your results also to clinical practice, because I think that if we try to take care of these results and have a look from different point of view, we can have a benefit from multiple disciplines. So I think that is one of the goal of this issue was obviously to try to stimulate several scientists, clinicians, and so on to have a look to this experiment, just to try benefit from this point of view. Alessandro, in the meantime, is preparing a list of questions that arrived in the...
Yes, Lydia and Christopher, we have prepared, we prepare some roundtable question, but I think that is more interesting to try to answer the question coming from the audience very numerously. And I will start, sorry for this boring going through the slides.
May I ask a quick question?
Yes, please.
Okay, so I had a question that came up during Alessandro's presentation. Yeah. So you were talking about the laser plasma accelerator?
Yeah.
Yes.
And I mean, obviously radiation physics is way outside my area of expertise. I have a background in biology, but I was intrigued because if I understood correctly, you said that by using that system, it's actually possible to create multiple different species of radiation simultaneously coming out of the plasma.
Yes.
So So the question I had, so NSRL basically, because they use a particle accelerator and then a system of electromagnets, even their GCR simulator isn't able to simultaneously expose either cells or animals to the full spectrum of GCR simultaneously. Basically, what you do is put the animal or the cells in, and they very rapidly switch between different ion types and sequentially expose them to kind of the spectra of what someone would see in deep space. Using your laser system, would it be possible to actually create like a multiple ion GCR simulator?
Let me say that what you are talking about is, of course, the last, the frontiers of the technology of linear accelerator created, let me say, specifically for this kind of space radiation simulation is in the US. I think that they started operation no more than 1 or 2 years ago, right? But of course, we are just, let me say, especially if we think that this kind of problem should be, let me say, there will be an exponential number of research and tests needed. It's the laser plasma accelerator could be a complementary technology, something that you can have in, let me say, in parallel with the standard linear accelerator technology, as the one that you mentioned. And that the idea is that it's not any radiation in a sequence. In that case, just because you produce the particles starting from a laser beam that hits a target, the target is a certain type of material, could be any different kind of material. You can, let me say, choose a target that produce at the same time different kind of particles. So the point, for example, could be very interesting if we want to simulate the Van Allen belt radiation. That is, as you mentioned, the Apollo mission, there was the NASA engineers or scientists that take care very carefully to avoid the astronauts most of the radiation. Very carefully they designed the flight path of the Apollo rockets through the Van Allen belts. Also, they try to avoid using the magnetic field of the Earth most of the problems. But of course, in the future, if we want to go to the moon continuously, or not in, we have to investigate more also the Van Allen belt space radiation. And so it's, let me say, possible, for example, to have from outside of the target electrons and protons simultaneously. That is the typical space radiation of the Van Allen belt. Most are not heavy nuclei, but are done by electrons and protons. So yeah, this is the, let me say, one of the advantage. Of course, the technology at the moment is, let me say, used for other purposes, but there is some facilities in Europe. There is a very big one in Romania, in Bucharest, in Magurele, to be precise. I was there in March to talk with the guy that under this kind of development, but also use of this kind of device. But I'm sure that there is other places also in the US that are using this technology. So it seems something very promising to have, let me say, complementary way to make this kind of tests that are very specific for space radiation, to mimic space radiation. Of course, first of all, for electronics components or for systems, but also for radiobiology. This is the... So I now move on to the, as I said, we pre-prepared some questions, but maybe it's better to try to answer the question from the audience. I will start from Lydia. If you can try to if you wanted to answer this question. I can interdisciplinary research involving radiobiology and other scientific fields contribute to advancing our understanding of radiation effect on living organisms, especially collaboration between radiobiologists and other disciplines like physics, chemistry, and genetics. I think that is make part of your everyday work, Lydia.
Yes, but I did also for Christopher. I'm very interested also to take care of the answer from Christopher on this regard. I think that is when we work in a institute like university hospital, we have the opportunity to work day by day with the clinician, biologists, chemistry, and to do several experiments to run, in particular several treatment, investigation, and so on. So we evaluate the opportunity to translate the knowledge from one field to another one. So I think that we can learn how, for example, the mechanism for the cancer induction that could be of interest for the space radiation, but it can be also necessary to be fully understood in clinical practice. We have also the need to know what are the dose constraints, the effect of different dose rate, the different spectrum that are incident on human. And we know that sometimes we have the need to study this effect using in vitro and in preclinical models due to the fact that the human is a very complex subject. So we have the need to try to simplify much more the knowledge and identify mechanisms that could be used just to not only to have the induction of disease, but just to block, for example, the carcinogenesis in a possible citizen. And we have a lot of ethical consideration when we run an experiment based on radiation biology. So Christopher is your main field because we take care of the animal reduction of the number of animals involved. We have like ethical committee, not only for the patient in hospital, but we have like ethical committee for animal use that is mandatory. So we take care of this aspect. But obviously, if you take a look, and we are discussing with several experts on ethics on this regard, and experts on animal welfare and so on, that we should think that these animals allow to better understand the clinical impact of a given drugs, of a given radiopharmaceutical. And without this clinical experiment, we are not capable to use directly in human new drugs and new components and so on, because this could be not so this could be not so ethical. But Christopher, may you try to share with us your point of view on this regard, on the ethical consideration and translation of research?
Certainly. So as far as the ethical considerations, I mean, certainly working with animals, there are a lot of ethical considerations that we have to consider, and we have to carefully design all of our experiments to make sure that we're addressing a reasonable question and there's good reason to believe that we'll achieve the outcome that we're looking for. I guess I would bring up another ethical consideration just theoretically speaking. So and it pertains to one of the other questions here, if we could look at differences in personalized medicine and things like that as a result of individual variation in response to radiation. This raises an interesting question that I know has been discussed as far as recruiting astronauts for different missions. So if we were to find out that there are certain SNPs or genotypes that are associated with an increased incidence or increased risk of cancer as a result of exposure to space radiation, one ethical question would be, what can NASA and the other space agencies do with that information? And what I'm referring to is, would it be ethically acceptable to genotype the astronauts and see whether they carry those particular SNPs and therefore might be at a higher risk of cancer, and as a result of that say you're not really suitable for the mission because it's highly likely that you'll get cancer as a result of that? Or what would you do with that? And I know I attended an online conference a few days ago that they were discussing at this point that NASA is actually getting DNA sequences from astronauts, but they're using that as a way of kind of predicting which ones may be more at risk of certain adverse outcomes. And I think they were talking about the effects on the eye, the ocular syndrome that some of the astronauts are developing as a result of being in space. But rather than excluding astronauts from missions, basically what they're doing with that is finding people that may be more at risk of developing those particular adverse events and making sure that they try to go in and prophylactically administer countermeasures to those people to prevent them from developing the adverse events. So, but I think that's kind of an interesting question as far as, I mean, personalized medicine and whether your genomic information should be available. And then, at least in the United States, that gets into what happens with insurance companies as well. If you have your genome sequenced and find out that you're at risk of multiple different diseases, what happens as a result of that information going out into the cloud?
Yes, because there are several concerns from the ethical point of view. Also, if prevent the possibility of we don't allow to astronauts to travel, but also if you apply this in daily practice, we should in this way, in some way, prevent also worker to be exposed to a given type of radiation, or you can prevent also patient to access to a given therapy. But if you take into consideration, for example, in clinical practice, in patients with BRCA 1 or 2, we avoid to use, for example, ionizing radiation just to avoid secondary cancer. And so I think that this could be a very nice topic to be discussed from an ethical point of view, but there are several aspects to be taken into consideration from citizen, from worker, from patient, and so on.
Yeah, I think that, Lydia, I would like that since it makes part of your direct experience, the last question could be very, very interesting, I think, for everyone, how this kind of research could be of help for the radiotherapy or radiation therapy for cancer treatment.
I think that day by day we noted that we are just changing the machine and the modality in which radiotherapy is performed actually in clinical practice. So if we have a very nice knowledge on the type of effect that could be so-called preclinical calculated or identified for the single patient, we can try not only to prevent severe and moderate to severe effects in the patient, but we can try also to generate treatment planning system, for example, that reduce the effect on lower grade of side effects induced by radiation. So I think that it's very important to know how several dose effect model, which is mandatory, just to have an idea on the better strategy to prevent not only severe effect, but also low-grade effect in patients. And at the same time, how to maximize, for example, the possibility to cure the patient. And this means that we learn from radiobiology that there is a very relevant impact of immune system response. So in some way, we can just have a benefit, for example, from these different disciplines on this topic, because sometimes we are just reducing the margin around the target treated for during radiation therapy, just to prevent the possibility to inhibit immune system and help us to cure the patient.
So unfortunately, the time is finished. So from the organizer of this very terrific conference, that I thanks to give us the opportunity to present our work. So we had to wrap up. Let me thank Christopher. Let me thank Lydia for their participation to the panel. I would like just to remind you that on this kind of topics, that it seems that raise a lot of interest. There will be also in the next future an interactive presentation and an oral talk at the International Astronautical Congress in Azerbaijan, Baku, in a few weeks, at the beginning of October. And especially I was very happy that a virtual work poster was accepted in the fall meeting in October 10-11 that is organized by the Lunar Surface Innovation Consortium, And okay, and then I think that we will present also somewhere else. Of course, you can contact me with this. There is the email, there is our website. I think that also Christopher could be and Lydia could be contacted or they can share the email address with everyone. And so 1 minute, no, 1 second left. Say goodbye. Smile. Thank you very much.
Thank you.
Thank you.